drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. Professor Amita Sehgal, thank you very much for joining me. I've wanted to talk to you for a while.
Well, good to be here. Thanks for asking me.
Do you want to give everyone just a basic overview of who you are and what your lab does?
Right. So I'm a professor of neuroscience at the University of Pennsylvania and an investigator of the Howard Hughes Medical Institute. I also head the Chronobiology and Sleep Institute at Penn. And what I work on is circadian rhythms and sleep at a very basic level, sort of drilling down into mechanisms, using primarily Drosophila models, although we now also use mice and a teeny bit of human work.
And so just for people who don't know, what is Drosophila and why do people work on Drosophila?
Right. So Drosophila melanogaster, which would be the more appropriate term, is the common fruit fly. And the reason that it is a great model for all kinds of studies, in my case, circadian rhythms and sleep, but people have used flies to study, to identify key aspects of development, how development occurs, how behaviors are regulated. And the reason it's great is because there's a lot of genetics. So genetic tools have been developed to study flies over many, many years. They are easy to breed and handle and relatively inexpensive. And it turns out that a lot of what we discover in flies is conserved all the way to humans.
Yeah, yeah, and I've had this conversation many times in my life, as I'm sure you have.
Right.
Fruit fly. It's not that people study fruit flies because fruit flies are just the most interesting organism, and we want to
Although
know
they are
everything.
that too.
They are. They are. They become interesting, especially after you start
Uh-huh.
using them. But
Uh-huh.
they breed quickly, and they've been very well developed as a model system,
Right.
meaning that scientists have all sorts of tools and tricks that have been engineered in the Drosophila model that really allow them to just drill down into amazing detail. And you can do things with fruit flies that you can't even do with mice or rats or other model organisms. And just to give people a sense, like, right, you can delete specific genes at specific times.
Right.
You can turn up a gene in a particular neuron in one part of the brain. You can look at the whole brain. You can see every neuron. It's all mapped out, every connection. You can really drill down into the basic biology in a way that you just can't do in so-called higher organisms. And as Amita just told us, as it turns out, we didn't know this several decades ago necessarily, but we know today that a lot of the core mechanisms in biology are conserved across animals. So in a fruit
Absolutely.
fly, in a human, things often work in a similar fashion.
Right. Right. And I think my field is actually a great example of that because the mechanism of the circadian clock, the internal 24-hour clock, was figured out in the fly. This is work that I was, very involved with. And it led to the Nobel Prize in 2017 in physiology or medicine. And that was because the mechanism found in the fly was not only conserved all the way to humans, but human sleep circadian disorders were found to be a consequence of mutations in genes that had first been found in the fruit fly.
Yeah, yeah. And even like when I got my start in science, I was in a Drosophila lab and it was
Really?
amazing. Yeah, I was in Sean Carroll's lab at University of Wisconsin.
Ah,
And
okay.
so I
Yep.
was studying development. But I mean, for those listening, because I was blown away when I learned this. And the scientists who did all of this work over many
Right.
years were blown away when all this happened in like the 80s and the 90s. When
Uh-huh.
we first started sequencing genomes and stuff, people thought, well, humans are going to have a gajillion genes and fruit flies will just have, a few genes. And development's obviously going to be completely different and nothing's going to be the same because flies are tiny and they have an exoskeleton and wings and we're these big complicated organisms. But as it turns out, a lot of the core genes and core molecules and mechanisms of development are remarkably similar between all of these creatures.
Yeah, and actually, one of the other things that's really surprising is not only are the flies tiny, but their developmental span is much shorter than that of a mammal, a human,
Yeah.
and yet the same genes are involved.
Mm-hmm.
So, one question that I know some people are interested in is how is it that the same genes operate on this time scale here and on completely different time scale in humans?
Yeah, yeah. And so with respect to Drosophila and sleep, first of all, can you just give us a basic overview here? Like, because people might not know this, some people will who are listening to this podcast. Drosophila sleep, what does that look like in a tiny creature like that?
Yeah, so they are completely immobile when they sleep. And there might be some twitching, a little bit of twitching, a little bit of grooming, but for the most part, there's immobility. That's one characteristic. They are less responsive to sensory stimulation, which is a hallmark of sleep. So, during sleep, you're less likely to respond to a sensory stimulus. And then if you deprive them of that sleep, and you can do this in different ways, they have to make up for the sleep they've lost. And, and so that means that there's homeostatic control of sleep. They need a certain amount of this rest sleep.
Mm-hmm.
And if they don't get it, they have to make up for it, which
Yeah.
is what is true in us too, right? Sleep is homeostatically determined, as is food intake, for instance.
Yeah, yeah. Can
Yeah,
you drill into that a little bit more? So when we think about, say, the circadian versus the homeostatic regulation of sleep,
right.
what exactly is the difference between those two things, and how are they similar and how are they different?
Yeah, so the circadian regulation of sleep basically refers to the 24-hour rhythm of sleep. We sleep at night, we're mostly awake during the day, although there might be an afternoon nap, especially in some cultures. But the homeostatic regulation is referring to the need for sleep that builds up often regardless of time of day. Okay. And so for instance, if you've been up all night, you will sleep in the morning, although your alarm clock and the circadian clock are telling you to wake up. And that is because there is a homeostatic need for sleep. We all have to get a certain amount of sleep. If we don't get it, we have to make up for it, even if it's against the circadian clock.
Yeah, yeah. And what does sleep in flies look like in general? Are they diurnal? Are they nocturnal? How much sleep do they need a lot of sleep or a little
Yeah,
sleep compared to
yeah. They,
us?
they actually do need a lot of sleep, and they are largely diurnal, but they do have a afternoon siesta. So what they have are like these morning and evening peaks of intense locomotor activity.
Yeah, yeah. And it's actually pretty common in diurnal animals, right? Like they tend to be pretty active earlier part of the day,
Yep,
later part of the day. And a
yep.
lot of animals, and humans included, have something we might call a siesta.
Well, I mean, there is this dip in
Yeah.
alertness in the middle of the day, right? And it is true that it is influenced by ambient temperature quite a bit. So in the warmer climates, the need is high enough that then you take an afternoon nap. In at northern latitudes, siestas aren't as popular, probably because the temperatures are lower. But there is a reason that the coffee carts have so much business in the afternoon.
Mhm.
It's because there is a slight increase in the homeostatic need to sleep.
Hmm. And so, the fact that flies sleep and humans sleep, worms, birds, everything in between,
Right.
it says that no matter what diversity we might look at, which is interesting, like maybe our brains when we're sleeping are doing something extra
Right.
or
Absolutely.
whatever that
Yeah.
aren't happening in a fly, but there's probably some kind of conserved core reason that sleep is seen like all over the tree of life. And because Drosophila are so distant from us in evolutionary terms, because they're so amenable to experimentation in detail, this is the model system where you can really drill in and start to ask, well, what is going on at the cellular molecular level that might tell us what the sort of core original reason for sleep is? Like, why do animals sleep at all?
Right. And this is actually why we developed a Drosophila model for sleep, right? So we did this about, I guess, 26 years ago. Um, and at the time, we had made a lot of advances in understanding the molecular nature of the circadian clock. How do you put a clock together? That clock, of course, we have to keep stressing, does not just regulate sleep. Almost everything in our body cycles with a circadian rhythm. Metabolism, blood pressure, body temperature. I mean, you name it, it cycles, all right? It's glucose, cholesterol, everything is rhythmic, which is why people should take time of day more into account than they do in life, in experiments, and in the clinic.
Yeah.
But anyway, that's for another day. So we knew that the fly had been a great model for studying circadian rhythms, and we asked, because of the advantages it had had, all the genetic tools and all that we discussed before, whether we could use it to study sleep. And so we asked whether we knew they had a rest state, because we used to monitor rest activity as a readout of the clock. And we asked whether that rest state is sleep-like. And it is, for the reasons that we discussed before, the arousal threshold, the homeostatic need. And the reason we really wanted to do this is, we are not trying to tell people that, as you just said, that fly sleep is exactly like human sleep and it serves all the same functions. I think with evolution, sleep may have taken on different, new meaning, for some organisms, but we feel like that some fundamental aspects of it, or the fundamental functions of it, let's say, must be conserved. And so clearly it's, otherwise it's different. So after we developed the fly model for sleep, people started thinking in terms of more small animal models. So they developed worms and fish, zebrafish. And some of them have these very interesting, unique features. Like, so octopus, for instance, has this pigmentation pattern that changes during sleep.
Hmm.
And that is thought to be the equivalent of rapid eye movement sleep in mammals.
Hmm. Hmm.
And so I think each model may have its own unique features, but yeah, the goal is to get at this very fundamental mechanism.
Right. And then obviously as a matter of common sense and experience, like if you ask someone on the street, like, why do we need to sleep? Well, we need to recharge, they might say.
Right,
We need
exactly.
to recuperate. There's clearly
Right,
something going on with energy.
right.
And so it's reasonable to suppose it's got something to do with core metabolism in our cells,
Right.
their ability
Right.
to make and utilize and harness cellular energy. And so, how do you start to get thinking about that as you go from just the basic experience of like, yeah, I need to recharge every day when I sleep. That's my experience, to starting to think about how to drill into that in a model system like the fruit fly.
Yeah. So I think, I mean, clearly, like you said, we all think we need to recharge, or, make our waking performance better, right? Why do we sleep? Because it makes wake better. And the question is, how does it do that? So, people have been thinking along the lines of, well, maybe during sleep, the brain stores glycogen. There was, 25, 30 years ago, this was a popular idea. There's glycogen. Could it be ATP? And it's not as simple as just building up more ATP. Okay, because there are things that happen during sleep that need ATP. There's, different regions of the brain where it might be regulated differently. And so we were in the fly doing unbiased genetic screens for sleep mutants. That was one of our approaches to finding sleep genes. We felt like that was the advantage of the fly. That really is what the animal lets you do, is do completely unbiased screens. And we felt like with the process like sleep, where so little was known, the unbiased approach was the best one. Like, you shouldn't start with some preconceived notions of what gene is involved and then, test it. Because if you do that, you're not going to get beyond what's already known in mammals. In fact, we sort of did that at the beginning, right? We said, do the neurotransmitters and neuromodulators that regulate mammalian sleep affect fly sleep? And the short answer is yes, they do. But so what? That's not getting you beyond what you know in mammals. And so we felt like, the fly lends itself to unbiased approaches, we'll take unbiased. So on the one hand, we were doing that. On the other hand, we started to, just think about what could be happening during sleep. And I had a couple of people in my lab, a grad student, a Greg Artushian, a postdoc, Paula Haynes, who came in with this idea that glia must be really important because metabolic needs of neurons are, facilitated by glia.
What are glia?
So glia are support cells, and neurons are the action cells in the nervous system. They are the ones that are, firing and driving activity, and glia are helping neurons. They're the support cells. And until recently, people used to ignore them. And now we are realizing they have a much more important role than we thought. Mm-hmm. And so they clean up, like, for instance, the extracellular space in the brain, because they may take up neurotransmitters that neurons have put out. But they are also providing nutrients to neurons.
Got it.
And my postdoc was very interested in how metabolic pathways are regulated over a sleep-wake cycle in neurons and in glia. And one of the things that she did, so one of the things about studying metabolism in the brain is there are not a lot of good assays to look in specific cells. There aren't good reporters, in other words. And one of the things that we could find a reporter for, and that she looked at, was to see whether oxidation in mitochondria was increased at the end of a day of wake.
Yeah.
Right? The idea being neurons are firing a lot during wake. They're exhausting their energy powerhouses, the mitochondria. So at the end of a day of wake, the mitochondria will be oxidized. And when she looked, she realized mitochondria were oxidized at the end of a day of wake, but not in neurons. They were in glia.
I see. Okay.
Yeah, they were in glia. And so we started to ask, like, why is that? And the oxidation in glia is as a result of neural activity. So we thought, okay, so somehow neurons are passing their oxidative damage.
Yeah.
Yeah, to these glial cells.
Yeah, it has to be happening in the neurons, like, because they're doing stuff. There's going to be oxidation. Metabolism needs to fuel all of the things they're doing. But somehow, instead of holding on to the stuff that gets oxidized, they seem to be passing that off to the glia cells.
That's exactly right. And we looked to the work of Hugo Bellin, who had shown in the fly model, but in the context of neurodegeneration, he was studying neurodegeneration in the fly eye. You can express genes that will cause neurodegeneration. And what he found was that the neurons were passing their damage to glia in the form of lipids, fats. So, these are oxidized fats that are now. Mm-hmm. So we thought, could it be that this is happening in a daily sleep-dependent fashion? And that was what we found, was that that the neuronal mitochondria were protected because the neurons were passing on oxidized lipids to glia. And and it is through the transporters, which people might be familiar with. It's the apolipoprotein D and E. So apolipoprotein E is huge in Alzheimer's because the E4 isoform is the highest genetic susceptibility factor.
Yeah. So ApoE, ApoD, there's other Apo
that's right.
things out there. And people will often hear about these in the context of cardiovascular disease,
exactly,
neurodegenerative disease. But these are structures that hold and move around fat, basically.
Yeah, they do. And so these are involved in passing these lipids from neurons to glia. And if you don't allow those fats to be transferred from neurons to glia, then the oxidative damage remains in the neuron, and the neuronal mitochondria get oxidized.
And what does that do to them?
So, in the long term, and at that particular time, we didn't look at it, but now we have mutants that have the same effect, and they're impaired in memory, they're impaired in lifespan.
Yeah, so you don't want these oxidized lipids building up in and near the mitochondria. It basically causes them to be dysfunctional.
Yeah, dysfunctional, exactly. Yeah.
So the idea is somehow, the neurons are doing their thing all day. They're sending signals, information is happening to create behavior and move the animal around. They are oxidizing fats. They are doing what mitochondria do with ATP. They're doing oxidative phosphorylation. So these oxidized lipids are building up in these little bubbles of fat, basically, these like ApoE, ApoD, the things that you mentioned. And somehow the neurons are set up to export these bubbles of oxidized lipids out of themselves and into these glial cells.
That's right. Yeah.
Now, do you know what happens next? Like, do the glial cells clean up the mess somehow? Like, do the glial cells get sick? What happens to these oxidized fats that are like otherwise going to cause a problem?
Yep. So that is the paper we published recently where we asked what happens to those lipids. And while we haven't directly tested it, we think that some of them are broken down in the glia via beta oxidation. So they generate some energy there. But the bulk of them are transported out of the brain. And so this is work done by a postdoc, Vamsik Cho, in my lab, where he was interested in the role of the immune system in sleep. Mm-hmm. So we know that immune molecules can affect sleep, although it may be most relevant for what we call sickness sleep, which is a whole different story. But sickness sleep is, the extra sleep we might engage in when we are sick.
Right. A lot of us have been sick before. You get the flu, you get a cold, you feel sleepy all day, and you sleep more.
And you sleep more, exactly. And the sleep is restorative, like it helps you recover from illness, but it is different from normal sleep. So that actually is an area of research in the lab, is sickness sleep. And we think that the sickness sleep is restorative for purposes of the illness, but it's not restorative in the sense of normal sleep. So, like normal sleep makes you perform better in terms of learning and memory. We don't think sickness sleep would do that.
So somehow they're both recuperative, but they're fixing different issues.
Yeah. So, we this is sort of going off on a tangent a bit, but my postdoc Sarah Noya did this work where she compared, this was in the mouse, compared different brain regions in terms of their metabolic activity, metabolites that build up in these as a function of sleep during sickness, normal daily sleep, and sleep following deprivation. And what she found was that the normal daily sleep and sleep after deprivation were similar to each other. They were energy restoring processes. You had more nucleotide triphosphates. Sickness sleep, on the other hand, was energy depleting. And so our interpretation of that was that during sickness, you're getting immobilized so that you're not expending energy on locomotion and other activities, so that the energy can go towards the immune system.
Right. Okay. So the basic idea here is normally I go to sleep at night and I wake up, I feel refreshed. I'm less groggy than I was at the end of my long work day. I am restoring my brain's ability to do learning and memory stuff, say, as one piece of what's going on in what we'll call normal sleep. But for sleep sickness, my body is saying, no, let's just immobilize ourselves and go to sleep. But instead of putting the energy towards the stuff we do on a normal sleep, we're going to take it and use it to fight off this infection and not waste it elsewhere.
Yeah, exactly. That's what we think is going on. And so if you look at, sleep in mammals is assayed by EEG, electroencephalogram. And when you look at mice that have been induced to sleep by giving them LPS, lipopolysaccharide, which is a component of bacteria. So you're giving them an infection, essentially. They sleep more, but that sleep looks very different.
Got it.
They have essentially no rapid eye movement sleep. It's not normal sleep.
Yeah. Got it. So they're asleep, they're behaviorally quiescent, they're immobilized, but what's going on under the hood is quite different.
Yeah. So anyway, so this was sort of my tangent from immune molecules are implicated in sleep, and I think they're more relevant for this type of sleep. And so in mammals, in flies, for instance, some years ago, we identified a sleep-inducing molecule that turns out to be an antimicrobial peptide, right? It's an immune molecule. I think it's relevant for sickness sleep. The immune signaling pathways that cytokines affect probably all are more relevant for sickness sleep. But so my postdoc, when he came in, was like, okay, but we do know immune molecules affect sleep. Do cells of the immune system affect sleep?
Right.
And in the fly, nobody's looked at that. And so in the fly blood, which is called hemolymph. So flies don't have blood vessels. They have an open circulatory system. It's like the organs are like bathing in this fluid.
Yeah, they're not pumping things through tubes like in us. It's like an open bath of fluid.
Right. Yeah, an open bath of fluid. And so they have hemolymph, but in that hemolymph, they have cells, just like our human blood has cells and it has molecules. So they have cells, and the major cell type is, you can call it a hemocyte, it's a macrophage. So it is an immune cell type.
It's like a white blood cell that we have that would gobble up bacteria or something like that.
That would gobble up bacteria, yeah. So these, so we started looking at these macrophages, hemocytes, and basically just to see, because most people who study them, in the abdomen, or they study them in developmental stages. So we started looking at whether they're even in the head cavity. And the answer is yes, they are there. And they seem to localize there specifically during times of high sleep.
So they go there, but they're not there all the time.
They go there, but they're not there all the time. And they're high during the afternoon siesta, and they're high at the middle of the night.
Yeah. Okay.
Yeah.
So macrophage-like cells, these are immune cells. Their role is to, gobble things up, basically. They're all over the fly, but they go into the brain, into the central nervous system when the animals are sleeping, which is midday siesta, and then, basically the nighttime when they're deep asleep.
Right. And they seem to dock, I guess that I always think of that word as being the most appropriate here, dock at the dorsal part of the brain, so a specific part of the brain. And and they're not quite penetrating the brain. They kind of seem to be more at the blood-brain barrier.
Yeah. And, obviously I kind of know where we're going a little bit, but an analogy I have in my brain is, I live in the city here, and the buses are always moving around, and they're transporting things around, and they have specific stops they make. And they're most active at certain times of the day, and they're less active at other times of the day. And then they go and dock at the bus station. So there's times a day where they have this pattern of where they go. And their whole purpose is to move around in order to transport stuff and to transport people. Or maybe we'll talk about a garbage analogy. The garbage man comes at a particular time of day to get rid of stuff that's piling up. What's the next part of the story here?
Yeah. So these are little buses, and they go to the brain times of sleep. And so we were like, well, why are they going to the brain times of sleep? Do they have anything to do with sleep? And so we thought, okay, well, the way to figure that one out is to target their major function and see if that affects sleep. And so their major function, as I was saying, is phagocytosis. They engulf bacteria. They also take up lipids, right? That's also, So they kind of take up stuff from outside. So we looked at all the proteins, receptors, and transporters that might be relevant for that particular function of these cells. And we knocked down each one of those proteins separately, individually. And then we looked at sleep. And what we found was that the knockdown of this one particular receptor, which is called EATR, and which, we did not name it. It has that name from before. When you knock down EATR, you have a reduction in sleep. Mm-hmm. In males and in females. And that's actually kind of important because a lot of things have sex-specific effects.
Right. So it's like you get rid of this gene or you knock it down, the animals sleep less. So either their ability to fall asleep is broken and or their need to sleep has been lessened. And so what is this gene doing? What does it do?
Right. So actually I'm glad you raised that point because that's also an important point. When you have a mutant that sleeps less, there's a big question about do they have less sleep need or can they just not sleep? And
right.
I think this would be in the latter category. They just can't sleep.
Got it. They can't fall asleep, but their need to sleep is still there. That would imply that
they fall asleep, but they can't maintain their sleep.
They can't maintain it. Okay.
So they keep getting up. Yeah.
Got it. So there's just less sleep. Sleep is disrupted.
Yeah.
What's going on at the cellular level? What is this gene doing?
Right. So we, looked at what the functions of this gene are. And so one of the things that it's involved in is taking up bacteria. And we didn't think that was relevant in this case because we're not infecting with any bacteria. Mm-hmm. So the other two things it does is it's involved in cell-cell adhesion.
Yep.
Right. And in uptake of lipids.
Got it.
And so we looked at whether the docking of the cells to the brain is when you don't have this protein. And there were fewer cells. So the other thing I didn't mention is we've actually also documented actual physical contact between these cells and the blood-brain barrier. So they actually physically contact it. Yeah. So what we think is going on is that without this protein, which is mediating adhesion, these cells cannot adhere to the barrier.
Right,
so they kind of fall off and they're not there.
Yeah, I mean, it's literally like it's like a boat docking at a dock. There has to be for the boat to stay there and for the cargo to get unloaded or loaded. You got to tie a rope around the dock.
Right. Yep. Yeah. It has to be moored. Yeah. And so it's not they're not there. And then we thought, okay, well, in our previous work, we did find that these lipids were building up in glia as a function of neural activity. So could this be involved in regulating lipids in some way? Because this protein is involved in lipid uptake. And so when we looked at the brains in these mutants, they had more lipid. Yeah.
So the lipid is building up in the glia, I'm guessing, and it's not leaving the brain.
It's not leaving the brain. It's building up in the glia. And we know that it's driven by these blood cells because we even did transplantation experiments, where we take this mutant, we put in normal blood cells and the animals have normal sleep.
Yeah.
So when you don't have it, there's more lipid in the brain. And there's less lipid in these cells because we also looked at the lipid in these cells.
Yeah.
And they also have some amount of lipid because that's one of the things they do is take up lipid.
Yeah.
And they had less lipid.
Yeah. So the story that seems to be coalescing here is the animal's awake throughout the day. Neurons are very active. They're very metabolically active. They're doing a lot of stuff. It's using up ATP. That's resulting in oxidation, including oxidation of lipids. Inside of these things, these little bubbles of oxidized lipids are building up. They're going to get exported somehow to the glia. And it sounds like, at nighttime, maybe these macrophage-like molecules come in and take out the trash from the glia and then
Take out the
remove
trash.
them.
Right.
But if you mess with these docking proteins and stuff that enable them to come in and dock where they need to dock, then the trash just keeps piling up.
That's exactly right. And so, yeah, the trash keeps piling up. So of course, so now we have this cell, which if you don't have this protein, these macrophages have fewer lipids. The glia have more lipids, but that doesn't really directly answer the question of, are they taking it out, right?
Yeah.
And so what we did then was to label the lipids accumulate in these things called lipid droplets. So we label the lipid droplets. And we were able to do that because lipid droplets also include proteins. And so you can label a protein with green fluorescent protein. You put a green tag on the protein, label the lipid droplet, and then we put that, transgenically express it only in the glia that normally build up the lipid droplets, which are close to the blood-brain barrier. And we put it in there, and then we realize that those green spots, now labeled droplets, end up in the macrophage, in the blood cell. And but if you don't have this protein in the blood cells, the eater protein, they don't end up there.
Yeah, because they can't go to where they need to go and then dock there.
Right.
So it does seem like kind of like the garbage man analogy. Like the trucks need to come in, they need to go where they need to go. They need to sit, it's like when the garbage man comes here, he needs to sit outside the garage for a minute and then, load the cargo.
Load the car, yeah.
And sometimes they don't come, right? Every few weeks there's a week where they don't come and there's a big pile of trash outside.
Right. Although I do think it's more than just the docking, because this protein is actually involved in lipid uptake. So it's probably not just the fact that the garbage man isn't coming. It's also that when the garbage man is coming, he's a little incapacitated in his ability to take out the garbage.
Yeah. Okay. And so what is the effect? So when you mess with this process and these oxidized lipids build up in the glial cells in the brain, what is the effect on the animal the next day?
Yeah, so it's huge, right? And this is building up, right, day after day, because we're looking at these animals over, weeks. And you got a lot of lipid buildup in the brain. You got a lot of oxidation in the brain, in mitochondria. And so there's, lots of bad things happening. But the other thing we discovered too, though, was we asked what kind of lipids are taken up by this protein and on this macrophage. And it's not neutral lipids. It's lipids that are either oxidized or lipids that are acetylated.
Right. So lipids that have been modified in some way and presumably modified in a way that makes them now garbage. They're toxic. They're harmful in some way. The cell wants to get them out of there.
The cell wants to get them out of there. And so we started focusing on acetylated lipids because acetylation happens on lipids, but it also a lot happens on what are called lipoproteins. So these are proteins that have, fatty acid conjugated to them. And we asked, so what is happening? So could it be that if this protein's not there, these acetylated lipoproteins are building up in the brain? Because it's easier to assay for acetylated lipoproteins than it is specific lipids. Mm-hmm. So we looked at acetylated lipoproteins and we found that all acetylated proteins in the brain are increased when you don't have this particular protein on the macrophages. And the increase in acetylated proteins, there are a lot of proteins now that are more acetylated that are building up in the brain. But the two key ones that we were interested in are called DRP1, which is something required for mitochondrial fission. It basically maintains the health of mitochondria. And the other one is PGC1 alpha, which is required for the synthesis of mitochondria. So acetylation of these two proteins really damages the mitochondria. Yeah.
So these proteins are essential for things like mitochondrial biogenesis
and function.
Yeah. So it's basically a way for the cell to track how impaired its ability to make more mitochondria, for those mitochondria to function, builds up. Right. And, this is a signal that says like, hey, like we're going to be in trouble if we don't get rid of this stuff.
Right. And so, yeah, so there's a lot of acetyl-CoA in the brain, which is a sign of metabolic stress. So these brains are under metabolic stress.
Right. Right.
And so there's more oxidation, and they are then, as a result, impaired in memory and shorter-lived.
Yeah. So if I try and just think about this in a kind of commonsensical way, when we sleep deprive ourselves because we stay up all night to study for an exam, or we were out partying or whatever, right? We feel more tired. We are mentally impaired. It's harder to remember things and to focus. And you go to sleep and then you are recuperated. Those problems go away. They get reset. You get re-energized, so to speak. What that could correspond to, based on what you're telling us from your experiments, is throughout waking, these oxidized lipids and things are building up. The ability of our mitochondria to create ATP and run all of the machinery of the cell is going down because all of this trash is basically built up. And what sleep is doing is literally allowing the trash to get cleared out.
Absolutely. Yeah.
One thing that's kind of interesting too, and it ties into some conversations I've had recently on the podcast in other contexts, which is, you said these are macrophage-like cells coming in, and in other contexts, they're going to respond to an infection. They're going to respond to bacterial endotoxin, and they're going to clear out infected cells. But in this case, that's not what we're talking about. So the immune system isn't just about fighting off an external invader. It's about recognizing problematic molecules and stuff that the body itself is producing intrinsically and taking out the trash, so to speak.
Oh yeah. I think, and macrophages in particular have a lot of endogenous functions.
Yeah. And I mean, what I learned recently is like even our bodies have, antibodies that will, bind to bacterial proteins, but in some cases, the exact same antibodies and other things will bind to lipid peroxidation byproducts, oxidized lipids, and trash, so to speak, cellular trash.
Right. And we think that these, so, in mammals, mammalian brains, you've probably heard about microglia.
Yeah.
There are like these cells that are involved that have immune functions, but they also take up lipids from neurons. So we think that the macrophages that we're looking at in the Drosophila circulation may be the evolutionary precursors of microglia. Yeah. Because Drosophila don't have microglia.
Yeah. So they're behaving kind of similar to microglia, but in us or in rodents, microglia, they're basically macrophages that live inside the brain at all times.
Yeah. So in mammals, microglia live inside the brain. The other cell type that might be analogous is the mammalian brain has these macrophages at the border, like right outside the brain, that are called border-associated macrophages, BAMs. And they might also be, I don't know, analogous to these.
I see. It's so those. I mean, I don't really know about those, but those, if they're right at the blood-brain barrier, that's sort of like, I don't know, that's like a bouncer at a nightclub. They're standing guard.
That's right.
Okay. So, what is sort of your overall takeaway from this recent paper about these macrophage-like cells that are coming in to take out the trash?
I mean, I think it sort of reinforces to me that sleep is, well, first of all, that sleep is involved in clearing the brain, and that there are multiple ways you can clear the brain. We've shown in the past that sleep promotes autophagy, right, clearance within a single cell. We've shown that sleep promotes movement of molecules across the blood-brain barrier via endocytosis. And now this is, another mechanism. The fact that it affects the periphery, because these are peripheral cells that are coming to the brain, sort of reinforces the idea that, sleep is not just of the brain, for the brain, and by the brain, as was thought to be the case, some years ago. The periphery is very involved in it. It raises to me the possibility that this is happening not only in the brain, but perhaps also elsewhere.
Yeah. And how does your work, the work you just described, fit in with things that other labs have been seeing over the last few years? Just to give people one example, in an episode I did recently that I'll link people to, that because I think it's nicely related to this episode with Gerald Miesenbach, they basically found a mechanism inside of critical neurons that control sleep in the fly, basically the neurons that make the fly sleep. Those neurons have this wonderful mechanism where they're basically counting and remembering how much of these lipid peroxidation products, these oxidized lipid things, are building up throughout the day. They're basically counting and remembering that. And then at some point, it triggers them to fire. And the basic idea is these neurons are saying, all right, now we've got too much lipid peroxidation. We need to make the animal sleep, presumably to clear that stuff out, which is kind of what you showed.
Right. Yeah. So I think that what Garo is looking at is the regulation of sleep by these specific cells, right? What we're looking at is more the function of sleep for the brain.
Mm-hmm.
And clearly we're thinking about similar processes. And I think the idea that the mitochondrial activity or lipid peroxidation is affecting sleep onset, which is what he's looking at, the regulation of sleep, and is also a function served by sleep. I think it makes perfect sense. So for many years, people used to break the two things down. It was, what regulates sleep, and then what does sleep do? And I don't think that those things should be really studied independently, because it sort of makes sense that whatever process is served by sleep, right, as a function of sleep, would be involved in inducing sleep.
Right.
If the process is broken, it's going to be sleep so that I can get fixed.
Right. Exactly. It makes perfect sense.
Yeah. And so I think that what we're looking at is how sleep is serving the brain. And then he's looking at these neurons that at the end are turning sleep on. We do think that sleep need actually can build up across the brain. Okay. And that maybe at the end that need is getting transmitted perhaps via lipids to these specific regions of the brain where sleep promotion more directly.
Yeah. I just want to drill in a little bit more for people. So when you impair the ability of certain cells to clear out the trash, as we've said, when these oxidized lipids build up, what goes wrong with the fly? I would imagine their learning and memory is impaired. I would imagine they're behaviourally a little wacky. Like, can you just describe that a little bit more?
Yeah, so their learning and memory is usually impaired. That is, sort of very much fits with the idea that sleep loss really results in. And they're shorter lived. Almost all the short sleeping mutants are shorter lived. And that's because, I should clarify, we were talking earlier about if you have less sleep, it could be because you have less need to sleep or because you have the need but can't sleep. If you have less need to sleep, we would not expect that you would be shorter. Right. But if you have the need to sleep but can't sleep, you're a classical insomniac. That is when lifespan is affected. And most of these mutants with the oxidation have the need to sleep, can't sleep, and are shorter lived.
Yeah.
Some of them do have a little, they're a little uncoordinated in their movements, but that we don't think is necessarily related to their sleep loss. We think it's more that particular gene is also affecting neural activity independently.
Yeah.
So like the shaker mutants,
Yeah. So again, the things I want to repeat for people that I think are important here are, the immune system isn't just responding to external invaders. It's also taking out the internally generated trash. Its general purpose is to get rid of bad stuff, basically. That might be a bacterial infection. That might be lipid peroxidation byproducts that are just building up as a consequence of oxidative metabolism. But the trash needs to be taken out and moved and disposed of, or it builds up, and that results in memory problems and behavior problems and all the rest. And the drive for sleep seems to be related to the buildup of these things. And somehow a major player here is these oxidized lipids that are problematic. The cells don't want them to build up.
Right. That's correct. Yeah.
So you've also studied another aspect of the cellular cleanup crew, let's say, another organelle that's really interesting that I haven't studied in too much detail. And those are peroxisomes.
That's right.
At a high level, can you just give us a sense for what do we know about peroxisomes generally?
Yeah. So, fats are usually broken down in mitochondria via a process called beta oxidation, and that, generates energy. But there are specific kinds of fats, like very long chain fatty acids, that go to this different organelle that's called a peroxisome. which aren't studied nearly as much as, mitochondria and other cellular organelles, but are really very important. And so there are specific things that get targeted to peroxisomes, but peroxisomes also have the additional function of getting rid of oxidative damage. And so they actually synthesize specific types of lipids that reduce oxidative stress. And, because of their dual role in oxidation and lipid metabolism, we became interested in whether these had anything to do with sleep. And so this is something that's on bioarchives. It's just a preprint at this point, but we found that peroxisomes do indeed build up with wakefulness. And we think that what they do is during sleep is process lipids and get rid of the oxidative damage. So they're contributors to this.
Got it. So one way to interpret that would be that as the animal's awake for longer periods, right? Metabolic activity is happening. You get these lipid peroxidation things happening, and you need more peroxisomes, therefore, to deal with that. And so that's why they build up as wakefulness progresses.
Right.
And so what happens if you mess with the peroxisomes and their ability to do what they do?
Yeah. So when we mess with the peroxisomes, you get more oxidative damage, and the animals sleep less. You can actually, treat them with an antioxidant, which will rescue the sleep. So there's oxidation that builds up also in the brain.
Yeah.
Yeah.
What antioxidant?
It's called NACA, N-acetylcysteine, I think.
I'll say so. Basically, you can mess with the ability of the peroxisomes to function. The flies, there'll just be more like lipid peroxidation and stuff in the brain. So the flies have problems, but you can supplement them with NAC, N-acetylcysteine, and that largely counteracts that.
And that largely counteracts that. Yeah.
Wow. So basically, it's just without the ability of the peroxisomes to deal with oxidized lipids, more oxidative stress happens in the brain, and it messes stuff up.
Exactly. Yeah. I mean, so, there are multiple components that come together to accomplish what sleep needs to do. On the one hand, you have the glia, and then the macrophages, and you have the peroxisomes within cells. So, yeah.
Yeah.
We also, I was mentioning earlier that we found sleep regulates autophagy. Mm-hmm. So that also is promoted by sleep and to clear up waste products within the cell.
Yeah. Is there any sense that, so you can rescue a lot of the effects that you just described by supplementing with an antioxidant. So the antioxidant capacity of cells is clearly critical. We're talking about oxidative stress here and how closely that ties to all of these things and why we sleep. What does that start to say about just antioxidant capacity and, what's in the diet, say?
Like what's in our diet, you mean?
Well, yeah, sure. But also just like the flies, like you said, you could largely rescue that phenotype by just giving them one antioxidant. That's pretty remarkable in my opinion.
Yeah. So I think that, in the case of the fly, you give them a bunch of this and you have some rescue. I think that I don't think that's very readily translatable in the sense that the amount of antioxidant that we would need to consume would probably be a lot.
So you're giving them like a lot.
Yeah. I mean, you've probably heard, people don't talk about it as much anymore, but some years ago, everyone was talking about how resveratrol, which is in red wine, kind of is this miracle drug that'll or miracle compound that'll clear up your oxidation and stuff. But the amount you would need for it to do that was just insane. Mm-hmm. Gallons and gallons of red wine every day.
Yeah. What are some of the other projects that you're working on in the lab that relate to sleep and particularly how sleep is interacting with mitochondrial metabolism and oxidative stress?
So one of the things that we've done, this is actually also a preprint on bioarchives, is to profile lipids in neurons and glia as over a day-night cycle and as a function of sleep to figure out which fats change which ones are more likely to be the ones that got transferred and which ones are actually promoting sleep. And so we've done that.
What did you find? I'll just say, my guess here would be that an important class of fatty acids that are going to relate to some of this stuff are polyunsaturated fatty acids.
The PUFAs.
Yes.
Yeah, so those are not directly sleep inducing in our hands. But I mean, they change, as do a lot. But there's a significance, like, because we're looking brain wide.
Yeah.
And we don't know yet. But on the subject of mitochondria, the other thing we found, and this again is a preprint on bioarchives, is that in a neurodevelopmental disorder, which is neurofibromatosis, the sleep loss is caused by a mitochondrial deficit. So this is, I don't know if you're familiar with the disorder, neurofibromatosis.
No.
Oh, okay. So it's, people know it. They used to call it, the elephant man disease. And people who have this have neurodevelopmental symptoms like, learning and memory and cognition and all that. They also develop tumors. They're called cafe au lait spots. They're benign, mostly benign, but they can also be malignant tumors. They are shorter in stature. It's actually, I think, a pretty common genetic neurodevelopmental, genetically transmitted neurodevelopmental disorder. And they have less sleep. And then there's a fly model, which really does capitulate a lot of the human phenotypes, and they have less sleep. And so we kind of tried to figure out what the basis of the reduced sleep was in the fly model. And there's a lot that went into this paper, but in a nutshell, it ends up being a mitochondrial enzyme that is very relevant.
And so you mentioned, glial cells. We talked about glial cells. They've been classically described as support cells. They provide certain nutrients to neurons. They're sort of sucking up and recycling neurotransmitters and things that leak out of the synapse and float away from the synapse.
Right.
And then they also have this role that you're describing where, they're basically the reservoir of cellular trash that can then get exported via these macrophage-like cells. So glia cells are it seems like over the last, 10, 20 years, glia cells have become much more important in terms of what we know that they're doing in the brain.
Right. They really have. I think they're sort of catching on. I mean, people used to, there were few people working on glia, and everybody else sort of viewed them with contempt, if I may say so.
Yeah.
Yeah.
And so somehow those oxidized fats get exported from neurons to glial cells. Do we know exactly how that happens? How those bubbles of fat actually get out of the neuron and get into the glial cells?
No, we don't.
Yeah.
We don't. Yeah. We know, which proteins, which carrier proteins are involved, right? That carry lipids. Hugo Bellin's work. So we haven't looked at this in the context of sleep-wake, but when he looked at it in the context of neurodegeneration, he found that there were specific cell surface receptors that were also involved, lipid receptors that were involved in this. But exactly how they are transported, we don't know. One of the things that people have asked me when I've talked about this work is, are mitochondria transported?
Oh, yeah.
Because apparently there are studies now showing that.
Yeah, I've talked to someone. Can't think of the name right now, but yeah, apparently they get transported between cells. They're in the bloodstream. They can travel far.
Yeah.
Interesting. So if we sort of back up and we think about all the work you've done and all the work that you've seen in flies and other animals, basically over the course of your whole career, what's your general take? So if you had to just not answer, but like the general question of what is this fundamental function of sleep? Why do animals need to become immobilized behaviorally and undergo what we call sleep? Do you feel like we're narrowing in on an answer to that question and it's sort of a fundamental metabolic function?
I feel like we are. I think that, I'm really excited that I think we are finally, getting a handle on this, right? And so I think that sleep serves to regulate metabolic, or in particular energetic homeostasis in the brain. That is really what it does. And it does that by maintaining the integrity of neuronal mitochondria. And how does it maintain the integrity of neuronal mitochondria? It does that at least in part by taking the damage away from those neuronal mitochondria, shunting it to glia, to blood cells. So that's clearly, one of the things it's doing, not to mention promoting other types of clearance. Stepping back a little bit, I started my career, my independent research career as a circadian rhythm researcher, right? So it was all about circadian rhythms. I knew nothing about sleep until we developed the fly model for sleep. And so I would say that even for circadian rhythms, the primary function is metabolic. That we evolved circadian clocks in order to segregate metabolic pathways and separate them in time.
Yeah.
And this actually, there's some, I think, really interesting work that had a big impact on me that was done by Benjamin Tu and Steve McKnight at UT Southwestern many years ago, where they showed that in yeast, which don't have circadian clocks, you separate oxidative and reductive phases of metabolism. They don't have circadian clocks, so this does not occur with the 24-hour cycle. It's more of a 3 to 5 hour cycle, I think, but you separate those, right? So I think that is a very basic thing that has to happen. There are metabolic processes that are incompatible with each other and have to be separated in time. In some algae, the circadian clock does this. You have to separate photosynthesis from nitrogen fixation. You can't have those occur at the same time.
It's just like a fundamental chemistry thing. They just can't happen in the same time, the same place.
It cannot happen at the same time, at the same place. And so the circadian clock, by putting things into temporal domains, it separates them.
Yeah.
And I feel like what sleep does, that sleep evolved in organisms that locomote. This is my worldview, right? This is my hypothesis, that in animals that locomote, sleep evolved. Because now when there's locomotion occurring during waking hours, the energy expended is so great that you need the other phase to be completely quiescent. And that's where sleep came in.
Yeah. And it makes a lot of sense, as you said before, that the regulation of sleep, homeostatic sleep need, and the regulation of sleep onset would be tied to, what sleep is doing functionally, because if you're building up some trash inside of cells, basically, that is the perfect signal to generate your sleep need, to measure your sleep need, to trigger the onset. And I mean, it just makes a lot of sense. And it is very reminiscent of things you see, like the circadian clock mechanism, all of the sort of transcriptional loops that you see that are involved in developmental processes, where basically you accumulate some product that serves as the signal to shut it off.
Right. Yeah. That actually, in circadian biology, we often talk in terms of the limit cycle, where, let's say there's a lot of a species on an island, a lot of prey, but then that leads to, when the predator comes along, it eats it all up, but then the predator now has nothing to eat, so it's gone, so then the prey grow again.
Right. Yeah, these boom bust cycles, and they have, I mean, you really do see it all over in biology. That's a lot of development is organized according to this type of thing, where you get the transcriptional loops. You build up a gene product, and then it shuts off its own production, and that type of thing.
Right. No, it makes perfect sense.
Yeah.
So anyway, I think, sort of getting back to your original question, I think that the clock and then the sleep also really serve to separate metabolic processes in time. And
yeah. And then I don't think we need to get into this, but it is interesting that you mentioned you have distinguished sleep for sickness purposes, for illness purposes, and sleep for, just normal sleep and waking. Like these seem to be redirecting energy for different ends.
Yeah. So I think that, and the other thing that we're interested in with sickness sleep, my colleague here at David Reason at Penn is developing a program, and that's in fatigue. So in the short term, sickness sleep is good. It helps you recover. But if it persists, then it contributes to chronic fatigue, and which is, a sickness state that could underlie things like long COVID and
Yeah. Interesting.
yeah.
Is there anything you want to reiterate from our discussion today about sleep in general, or any final thoughts you want to leave people with on this topic?
I think, I guess I will just say when we first developed the fly model, there was quite a bit of resistance to it because sleep researchers they were studying mammals or maybe some birds. And as you probably know that, birds sometimes sleep with one half of their brain, right? And the idea that anything less than that was sleeping was not acceptable to them. And I'm really happy that we've now gotten to a point where they're all talking about it, they're working on it, and like I said, I think there is some agreement, at least among some of us, on what the function might be. And there will still be people who will say, "Oh, it's not important for metabolism, it's important for synaptic plasticity, or it's important for memory." And my answer to that is we're not in disagreement.
Well, yeah. But I mean, those things are very energy intensive.
But you'd be surprised because in the sleep field, there are a lot of like psychologists who don't have much cellular or molecular background. And I was with one on a panel one time, and I said, I thought the function of sleep was metabolic. And he said, I don't agree. I think it's memory. And I said, I'm totally fine with that. But the question is, how does it facilitate memory?
Right.
And so we think that all of it would come down to this metabolic function.
Yeah. I mean, there's no way learning a memory can't be affected if you're
plasticity.
affecting. Yeah.
In fact, we find that even the role of sleep in memory, which we've also worked on, is to some extent dependent on the metabolic status of the animal.
Right. I mean, that makes perfect sense. I almost can't imagine how it would be otherwise.
Yeah. So we find, like in an appetitive learning paradigm, right, where you are pairing sucrose with a smell and teaching the animal to make that association. If you feed the animals after they've been trained, then they need sleep to consolidate that memory. If you starve them, they don't need sleep. And we've actually mapped the circuits for those. They're parallel circuits in the fly brain that drive that different types of sleep. And the different types of memory, I'm sorry.
Well, thank you very much for your time.
Sure.
I've been following your work for many years. So you're one of my favorites in the fly field. So I encourage people to look at your lab. So I'll link to some similar episodes and some of your work in the episode description on Substack. And thank you again for your time. Really appreciate it.
All right. Thanks for having me. I really enjoyed that. That was fun. Thank you.
Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water. Heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.